The closer the two were in sequence, the closer the double helix's melting point would be to the perfect match standard of 95 degrees centigrade. The closeness observed was surprising indeed. King was able to infer that human and chimpanzee DNA differ in sequence by a mere 1%. In fact, humans have more in common with chimpanzees than chimpanzees do with gorillas, the genomes of the latter two differing by about 3%. So striking was the result that King and Wilson felt obliged to put forward an explanation for the apparent discrepancy between the rates of genetic evolution, slow, and of anatomical and behavioral evolution, fast. How could so little genetic change account for the substantial difference we see between the chimpanzee at the zoo and the species on the other side of the glass? They suggested that most of the important evolutionary changes had occurred in the pieces of DNA that control the switching on and off of genes. This way, a small genetic change could have a major effect by changing, say, the timing of the expression of a gene. In other words, nature can create two very different-looking creatures by orchestrating the same genes to work in different ways. The next and biggest bombshell from Wilson's Berkeley lab came in 1987. Using patterns of DNA sequence variation, he and his colleague, Rebecca Cann, figured out the family tree for our entire species. It was one of the very few pieces of science ever to make the cover of Newsweek. As Krings would in his analysis of Neanderthals a decade later, Cann and Wilson relied on mitochondrial DNA. There were several reasons for using mtDNA, but as usual, the practical ones were most important. In the days before PCR technology had entered the research mainstream, getting enough DNA to probe a particular gene or region could be quite a headache. And Cann and Wilson's study called for analyzing not one, but 147 samples. They therefore needed as much DNA as they could get their hands on. A human tissue sample is massively rich in mtDNA compared with the chromosomal DNA found in cell nuclei. Still, Cann and Wilson would need plenty of tissue if they were to have any hope of extracting even mtDNA in sufficient quantities. Their solution? Placentas. Usually discarded by hospitals after babies are delivered, these are a rich source of mtDNA. All Cann and Wilson had to do was persuade 147 pregnant women to donate their babies' placentas to science. 146, actually, because Mary Claire King was more than willing to contribute her daughter's placenta. And they knew that to reconstruct the human family as completely as possible, they would need tissue from the most genetically diverse range of donors they could assemble. Here America's melting pot population offered a distinct advantage. They would not have to travel to Africa to get hold of African DNA. The slave trade had brought African genes to our shores. But Cann and Wilson would have to depend on collaborators in New Guinea and Australia to find Aboriginal women, not much represented in the U.S. gene pool, who were willing to participate. Your mtDNA is inherited from your mother. Your father's genetic contribution, contained in the head of a single sperm, did not include mitochondrial material. The sperm's DNA is injected into an egg cell that already contains mitochondria derived from the mother. Cann and Wilson would therefore be tracing the history of the human female line. Inherited from just one parent, mtDNA never gets an opportunity to undergo recombination, the process by which segments of chromosome arms are exchanged, so that mutations are shuffled from one chromosome to another. The absence of recombination in mtDNA is a major advantage when we come to reconstruct the family tree based on similarity of DNA sequences. If two sequences have the same mutation, we know that they must be descended from a common ancestor, in whom that mutation originally arose. Were recombination occurring, however, one of the lineages could have acquired the mutation just recently through a recombinational shuffling event. So having a mutation in common would not necessarily indicate common ancestry. Now the logic for using mtDNA to make the family tree is simple. Similar sequences, those with plenty of mutations in common, indicate close relationship. Sequences with many differences indicate a more distant relationship. In visual terms, close relatives, those that derive from a relatively recent common ancestor, will cluster close together on the family tree. Distant relatives are more spread out because their common ancestor is relatively far back. Cahn and Wilson found that the human family tree has two major branches, one comprising only various groups within Africa, and the other consisting of some African groups plus everyone else. This implies that modern humans arose in Africa. That is where the ancestors common to all of us lived. Well, this idea was hardly new. Noting that both our closest relatives, chimpanzees and gorillas, are native to Africa, Charles Darwin himself inferred that humans had evolved there too. The most striking and controversial aspect of Cahn and Wilson's family tree is how far back it goes in time. By making a number of simple assumptions about the rate at which mutations accumulate through evolution, it is possible to calculate the age of the family tree, the time back to the great-great-great-grandmother of us all. Cahn and Wilson came up with an estimate of about 150,000 years. Even the most distantly related currently living humans shared a common ancestor as recently as 150,000 years ago. Like Sarich and Wilson's result 2 decades earlier, Cahn and Wilson's was greeted by many in the anthropological community with outraged disbelief. One widely accepted view of human evolution held that our species was descended from individuals who left Africa about 2 million years ago before settling throughout the Old World. Such a model implied that the family tree should be about 13 times deeper. Cahn and Wilson's alternative, dubbed by the media the Eve hypothesis, or less misleadingly, out of Africa, did not deny the more ancient migration, but rather implied that when modern humans arrived in Europe, they displaced those populations of early hominids derived from the original exodus